second or third the statement of thanks to the organizers. Melanie for this session, Felisa, Morgan, I think Ethan, and several others did an amazing amount of work behind the scenes to pull this all together. I'm in awe of everybody here in this room. It's great to see everybody again. Jim, thank you for everything. It's been an amazing ride. Okay, so what I wanted to do today is actually go back and talk about something that actually really influenced me about Jim. And I don't know if you know this or not, but actually he's an evolutionary biologist. And you may say, sure, I knew that. But no, I really mean that because I think it permeates his thinking deeply. And it influences the entire research program coming out of the Brown Lab. And you may say actually that, well, you know, that should be expected, that everybody should at heart be an evolutionary biologist. But what I wanted to put in the title was actually Jim Brown is a revolutionary evolutionary ecologist. And this is perhaps maybe a better picture of what Jim is. And I'm going to actually emphasize 2 points why I think that he is a revolutionary evolutionary biologist. And the first actually comes from in taking several of his classes, community ecology, biogeography, let's see, the Kino class. At several points, he would turn and ask the students, in particular in the biogeography course, which was at undergraduate level, he would ask the students, how many people have read The Origin of Species? And there would be this awkward silence in the room, and a few hands would go up kind of nervously, and you kind of know, well, maybe they didn't read the book, right? And I have to say that, you know, I kind of skimmed it a little bit, but I hadn't really read it. But after hearing him say this over and over again in grad school, I finally did read The Origin. And it was truly remarkable and humbling because many of the things that we're working on, you realize, well, gosh, you know, Darwin already wrote about that a while ago. So, and to this day, I ask my students that question, and I get the same response, this scattering of little hands that go up. And so Jim really emphasized this, I think, to all of us. And so I want to talk about this work that we started back in 1995, and I have to say that in thinking about this at the time, if somebody would have asked me if I'd still be working on allometry, almost 20 years later now, which is hard to believe, I would say, gosh, you have to be kidding. But this work has actually opened my eyes in a lot of ways, and has forced me to think about the world completely differently. In particular, this is kind of the focal pattern, right? You know a lot of these different plots. All right, so this is body size of an organism versus its metabolic rate. And so here, this is for mammals. This is a paper that Van Savage headed up. Right, so we get a nice scaling relationship. These are interspecific scaling relationships across species. So this is for mammals. Here's for plants, some data that we put together. I put together with Carl. And again, in each one of these different plots, right, we get this approximate 3/4 power scaling and metabolic rate, right? That's going to intrigue people for a very long time. And so the perennial question here is, of course, why quarter power scaling? Why do we have a slope on log-log axes that's 3/4, right? Well, I'm actually more botanically oriented. And there's another pattern that's actually very interesting in the plant world. And a lot of the botanical ecologists have kind of rallied around this scaling relationship, and this is called the leaf economic spectrum. And what this is a relationship between the photosynthetic rate of a leaf, how much nitrogen is in a leaf, the LMA, which is the inverse of SLA. This is the leaf mass per unit area. It's a measure of investment. We can also put leaf longevity on here, and we get these global trait scaling relationships between these different plant traits. And what this is reflecting mainly is the classic life history, slow, fast continuum with plants that have either fast turnover times or slow turnover times. These traits are associated then with this basic life history trade-off. Right? And so one of the questions here that I want to really ask is return back to this original scaling work and ask what is the evolutionary basis for metabolic allometries. And when I say evolutionary basis, really the microevolutionary basis. And begin to ask the more pressing question, can we more formally integrate metabolic theory with evolution by natural selection? And so the overall goal here is I'm going to give you a kind of very quick overview of trying to test the core evolutionary assumptions of metabolic scaling theory. And it's actually very interesting. We can actually go back to the origin and ask, well, did Darwin have some of these little bits? Okay? Well, Darwin didn't really talk about allometry. The term allometry wasn't coined until Huxley. But if you go through and actually read then the origin, there are these tantalizing little quotes that Darwin kind of knew that these patterns were interesting. So here, page 11, chapter 1, this is variation under domestication. He says there are many laws regulating variation. Okay, he says laws regulating variation. I will only allude to what may be called correlation of growth. Breeders believe that long limbs are almost always accompanied by an elongated head. Right? And so this is as an organism grows during its ontogeny, we get this proportional changes in different parts of the body. And he says here, owing to the mysterious laws of correlation of growth. Right? A little later on, chapter 5, laws of variation, we can start looking into why these traits then covary with each other. And so he talks about, again, in laws of variation, in this subsection known as correlated variation. He says, I mean by this expression, that correlated variation, that the whole organism is so tied together during its growth and development, that when a slight variation in any one part occurs, they're accumulated through natural selection, other parts become modified. Right? So all of these different traits during growth, during development, are so intricately tied and covary with each other. And he says here, this is a very important subject, most imperfectly understood. So what about kind of the origin of these laws of correlation? Well, a little later on in the chapter on laws of variation, in the subsection compensation and economy of growth, you can see this almost an argument of optimality. In fact, throughout the origin of species, he uses the term economy in many different ways. Economy of nature, economy of growth, economy of how traits are related together. And so he says here, natural selection is continually trying to economize in every part of the organization. If under changed conditions of life, a structure before useful becomes less useful, its diminution will be favored, for it will profit the individual not to have its nutrient wasted in building up useless structure. Right? So there's this economy of resource use in terms of how these traits are related. And so the second point, what I want to say about why Jim is actually a revolutionary evolutionary biologist, is that he has consistently, in my opinion, pushed for a more deeper definition of evolution by natural selection. And so this is a paper that he published with Pablo Marquette in 1993. I actually remember this day very fondly because Jim, acting as if a new baby had been born, was proudly walking down the halls of Castetter Hall with a pile of reprints from AmNat, handing them out to anybody who would take them in the hall. And so the title is Evolution of Body Size, Consequences of Evolutionary, an Energetic Definition of Fitness. And so in this paper, they build on two quotes that stemming right after Darwin. One here by Boltzmann, the physicist, who states that the struggle for existence is a struggle for free energy available for work. Again, trying then to equate evolution by natural selection with the energetics of resource use and distribution. The second quote here from Lotka, the struggle for existence. In the struggle for existence, the advantage must go to those organisms whose energy capturing devices are the most efficient in directing available energy into channels favorable for the preservation of species. Okay. Again, this is from Boltzmann and Lotka. And so then Jim, Pablo, and Mark, they basically took that and defined then fitness as, fitness is defined here as the rate at which resources in excess of those required for growth and maintenance of the individual can be harvested from the environment and used then for reproduction. And so this is 1993. And at the time I thought that this would just really take off and really excite people about this link then with evolution by natural selection, then with energetics. And I was actually a little bit more disappointed. If you follow the trail of this paper, there's all this debate with Kozlowski basically coming down pretty hard on, no, come on, you cannot define evolution by natural selection in terms of energetics. Okay, we already have a proper definition of fitness. Why then the need to energy? So let's then fast forward here to 1999. So the argument for the origin of allometric scaling and quarter power exponents is basically building then on this argument in terms of linking then energetics then with natural selection. And so the idea here, and this is a quote saying that we conjecture that organisms have been selected to maximize fitness by maximizing metabolic capacity, namely the rate at which energy, material, and resources are taken up from the environment and allocated to some combination of survival and reproduction. And so the argument then for the origin of these quarter power allometries, the evolution of these metabolic allometries, is that basically natural selection has been acted to, on the one hand, maximize metabolic capacity, but at the same time minimize distances over which materials are transported within the body. In other words, the time required for transport. Okay, and so another statement of this paper is actually more far-reaching. It says that quarter power scaling relationships should apply to all organisms that have been selected to maximize metabolic power under the constraint of minimizing internal transport distances. So these are the statements, okay? And actually in many ways these were conjectures in the paper, okay, that selection then should be acting in this way. And so then my question is, where's the proof for this, okay? Can we actually show that then selection, right, can actually act like this? So the explicit evolutionary assumption is that competition between individuals for limiting resources then is resulting in a phenotype that is a scalable phenotype that can optimally uptake and flux resources or energy. The implicit evolutionary assumption is that the origin of quarter powers is primarily at the intraspecific level, right? Because this is where then selection is going to be operating. Not at the interspecific level, and those plots that I've showed you before are all intraspecific. So the idea is that selection can and primarily operates on allometric scaling exponents. That is, the exponent, the slope then, should be a trait. But then also, there should be strong stabilizing selection then on these quarter power exponents. Okay, so for the rest I'm going to tell you, I'm going to try to highlight a few bits of evidence here for trying to test the evolutionary basis of metabolic scaling theory. In particular, highlight intraspecific variation, not only these economic traits, but also these scaling exponents. But also ask, is there actual heritability in these economic traits and these scaling exponents? But also, is there a genetic basis of economic traits and scaling exponents themselves? And to actually answer these questions, I'm going to look at this lowly little weed that's known as Arabidopsis. And Arabidopsis is great because now there is a huge network of biologists and various technicians that have not only mapped the genes then in this model organism, but enable you then to go from the genes then to the phenotype. And so before you say it, I have now been seduced by the dark side of the force. The other side of the department has been calling, and Tom, maybe that's you. I don't know. This actually stems from when I was on sabbatical and I met a group of plant geneticists at the University of Montpellier. And over some nice meals, we began talking, and they came to my talk about metabolic scaling. And at the time, my postdoc actually spent time in both France and Arizona. Cyril was a really good intermediary here. And we started a collaboration with Francois, Dennis, and Christine in this wonderful facility that they have, that they've just built. It's basically a huge Arabidopsis factory. And in this factory, you can do amazing things in terms of measuring phenotypes and altering environments. And so for the methods is that we analyze the genetic variation in leaf economic traits, but also the scaling of plant growth. And we assess then the traits and allometry of two wild-type populations of Arabidopsis. Actually, one type coming from basically central Poland, and the other population coming from the Cape Verde Islands. And these are very different, climatically different places, but still same species of Arabidopsis. And so we're going to then grow not only the native populations, but also 120 recombinant inbred lines then between these two populations. And so from that, we also looked at some near-isogenic lines and some targeted mutants and did some QTL mapping based on these metabolic traits. And so this is actually a really fun facility because they have all these robots in these growth chambers that not only can measure then all these different plants, but they go through and the robot weighs the plant, they know how much soil is in it, you can weigh the masses, you can then alter how much water then is supplied, the robot takes pictures, measures soil moisture, you can do these experiments. So this is a rainout experiment, so this is a drought experiment. You can change daylight, you can do a bunch of things, and then the technicians of these facilities, then they have a specialized flux container, so you can then measure CO2, water fluxes to then measure for each individual plant, then the whole plant, and not only growth rates, but also CO2 fluxes, and so on. And what was actually interesting when we went there with these questions, this was amazing, they had actually never measured whole plant flux, okay, or whole plant respiration, and some of these other kind of standard measures that we use for these allometric studies. And so it's fun kind of trying to figure out methodologies for this. And so what I want to show you is that actually this just reiterates the point that Tom made. Within Arabidopsis, when we take these recombinant inbred lines, and so we take these two populations and we do these crosses, and so we allow then all these progeny then from this cross then to grow up. Okay, so there's no mortality here, so we just let all these individuals then grow up and we measure their masses, their growth rates, their nitrogen concentration, photosynthetic rates, age at flowering, and leaf mass per unit area. This is the variation in these traits in the recombinant inbred lines. The red lines here are the parent populations. So, and just one cross then between these parent populations, allowing then these seeds to grow up, we get this huge range in trait values. And I'll say that the range in nitrogen, LMA, photosynthetic rate, within Arabidopsis, this is a half of the global variation in these traits, all within just Arabidopsis, which to me is quite amazing that this little plant can generate that much trait variation. So what does the allometry of resource use look like? This is ontogenetic allometries then for each one of these various recombinant inbred lines and these parental types. And so here to make this easy, I'm showing two different parental types and then two different extreme recombinant types. This is then dry mass and growth rate. So this is then the scaling of their allometry. And actually this is significantly curvilinear. Okay, it's steep here and then it shallows out. But when we go in and we do more detailed analysis, that curvilinearity is solely due to the fact that we're crossing then these different lines. When we then keep the genetics then the same and look at their ontogenetic allometry, what we actually find is that for the two parental populations, the scaling relationship is indistinguishable from 3/4. But then when we look at the extremes of the recombinant inbred line, we get a slope of about 0.9 to a slope of about 0.6, which actually brackets quite a bit of the variation. And so this is just then showing that for the two native then populations, okay, our best estimate of the allometric exponent is actually centered around 3/4, but the recombinants then can then bracket in both shallower and then steeper than exponents. Okay, and so within these RILs we can then generate these crosses that significantly then deviate then from 3/4. That is, there's genetic variability then for 3/4. Interestingly, also within Arabidopsis, this is the leaf economic spectrum, that same correlation I showed you just within these recombinant inbred lines. They segregate out nicely. The functional form of this is actually exactly the same as the global leaf economic spectrum, but again, we're seeing that here within Arabidopsis. And are these traits heritable? And so we calculated broad sense heritability for not only size then at reproduction, but growth rate, the scaling exponent, age at flowering, photosynthetic rate, leaf mass per unit area. All these traits have high heritability, including the scaling exponent itself. And what's really very interesting, and this surprised all of this, is that when we did the QTL then analysis, for not only the scaling exponent, but also these life history traits like age at flowering, leaf mass per unit area, photosynthetic rate, nitrogen concentration. All of these different traits are then centered on two separate loci. Two separate loci then control all of the variation in not only these leaf economic traits, but also the metabolic scaling exponent. And so these are actually, the likelihood score here is actually quite significant, it turns out. These are very interesting genes that code for blue light receptors and this other transcription factor that has this very big kind of impact on the basically the size and shape of growth then of these plants. And what's also very interesting then is that depending on the trait, you can have either a positive or a negative score, and that just indicates whether or not that trait is negatively or positively then associated then with that loci. So then to conclude, so a simple cross of two genetic variants in Arabidopsis generates a large fraction of the global variation in the leaf economic spectrum, but also in metabolic scaling theory. And what's interesting is that these genes are actually these two antagonistic and they're pleiotropic. That is, one gene is influencing then these multiple traits. And these appear to be tightly linked to the control of the overall plant life history. That is, time until flowering, how fast you grow, the scaling of your growth, which is actually quite interesting. And so then both of these genes then appear to constrain the space of possible trait values. And in particular, you can't just select on one trait, because a change in one trait then leads to this cascade of change in all these other traits. Now, while I haven't shown you that you can select necessarily on metabolic scaling exponents, all these results are consistent with the general argument that these scaling, botanical scaling relationships are the outcome of natural selection. And so with that, I'll stop. But say thank you, Jim. The Brown Lab, past brownies, the Astro Brownies, a new term that I've learned today. And with that, if there's any time, we'll take questions, but probably not.